I'm Sean Hanlon from the National Cancer Institute, and also a member of the Generation of Human Tumor Atlases implementation team. And I'm going to be giving you a brief overview of the Human Tumor Atlas Network, or HTAN. So the concept of building tumor atlases was identified by multiple Blue Ribbon panel working groups in 2016, and HTAN is the realization of those ideas. The overarching program goal is to construct dynamic 3D atlases of human cancers. Specifically, these atlases should integrate molecular, cellular, and tumor tissue composition and architecture data, as represented in the figure in the right and the top panel. The atlases also describe key transitions in cancer, such as pre-malignant lesions to malignancy, or locally invasive to metastatic cancer, as shown in the middle panel on the right. The atlases focus on high-risk cancers, including those responsive and non-responsive to immunotherapy, such as which is represented in the bottom panel on the figure, and also pediatric cancers. These atlases also aim to represent a diverse patient population. And in the end, the completed atlases should enable predictive modeling that will help us to do things like better refine therapeutic choices for patients. This slide shows an overview of the HTAN consortium, which is represented in the figure on the right. The funded projects cover a total of 11 pediatric and adult cancer types. There are 10 HTAN research centers, which are evenly split between pre-cancer and established tumors. And there are also 2 pilot projects. Our moderator today, Dr. Li Ding, is the contact PI for the Washington University HTAN Research Center, and I've highlighted on those efforts on the slide in the red boxes, and they'll be developing pancreatic and breast cancer atlases. And our speaker today is Dr. Christina Curtis, and is a member of both the Duke University and Stanford University pre-cancer atlas teams, and I've shown those in the boxes in blue on the right, and they'll be developing breast and colon pre-cancer atlases. HTAN also includes a data coordinating center, which is tasked with developing and implementing data standards, storage, and sharing of data with the broader research community, as HTAN is really a data generation resource, as well as an investigational program. And relatedly, HTAN is preparing for our first major data release. And the data portal, which is shown in the figure here in the center of the slide, will launch with an initial data release during the May 17th to 21st AACR meeting that's coming up in a couple of months. And this will include a presentation from the HTAN DCC team during that during that meeting, with the exact day and time still TBD. But this initial data release will include both single cell and bulk sequencing data, proteomics data, and imaging data from several hundred tumor samples across 8 tumor types. So I'd encourage people that are interested in accessing that data to tune in and hear more about that data release. And with that, I would like to turn things over to our moderator, Dr. Li Ding from the McDonald Genome Institute and Washington University School of Medicine.
Thank you, Sean. And it's my great pleasure to introduce Dr. Christina Curtis for today's presentation. Christina is an associate professor of medicine and genetics at Stanford University. Her recent work focuses on quantifying the dynamics of tumor growth and resistance. So today, on behalf of the Human Tumor Atlas Network, Christina will present her work on spatially resolved single cell atlas of tumor initiation. Christina.
Thank you. So it's really a pleasure to present this work and work of many colleagues who have contributed to this important effort. And today, as has been described, I'll share our work to characterize initiation in 2 common cancer types. So over the years, my lab has had a long-standing interest in charting tumor evolution and molecular events through space and time. And we've focused on this in both breast and colon cancer, largely in advanced disease, using a variety of different molecular techniques, starting initially with multi-region DNA sequencing and moving increasingly towards single cell, as well as spatially resolved in situ proteomic and transcriptomic technologies. And much of our goal in doing this has been to understand the evolutionary dynamics of disease progression. And so just to provide an example, we're often starting with an established cancer and tracing back to the most recent common ancestor or the evolutionary history of the tumor. And while we can often get back to the founding malignant clone, it's far more challenging to trace back beyond this point to the precancerous lesion or even the zygote. And that's because typically most events are clonal once we've founded this clone. And so there's an important need to study earlier stages of disease progression. Now, we often view the genomic data as really the very first starting point for the types of questions we're interested in. And those data provide a readout of the natural history of the tumor because with each cell division, we faithfully record somatic alterations and the evolutionary lineage. Now, typically we build on this by also thinking about how to simulate spatially resolved tumors in 3D, keeping track of the relationship between individual cells. And then we couple this with powerful statistical inference techniques to infer back parameters of interest. And so this is something that's we've proven to be quite powerful to address a variety of questions, including those of a clinical nature. And for this reason, I was really thrilled with the announcement of the HTAN effort, as we heard about from Sean. And it's clear that understanding evolution and molecular changes is going to be critical for the development of improved predictive models. And so this really, it also became apparent that it was going to take a consortium effort of this kind if we were going to have the kind of detailed understanding that's needed, spanning the molecular all the way up to the cellular and structural and individual patient levels using a variety of different assays. And I'll touch on many of these different approaches today. So really, I'm going to focus heavily on the critical need for a precancer atlas of multiple tissue types. And this is because it's been seldom studied. And in fact, the earliest events during tumor evolution are poorly characterized. We have very limited understanding of what they are, in part simply because we haven't sampled these lesions for practical reasons. I also want to point out that because precancers are non-obligate precursors to invasive disease, they provide a very unique vantage point for studying the changes that can lead to some life-threatening cancers when they go on to metastasize. And really, understanding these events may provide us a new approach for both risk stratification as well as for intervention and prevention. And of course, this is at a time when it would be most effective. And indeed, work from my own lab has shown that there is a critical window of opportunity to capture and understand these earliest events, perhaps before the horse is out of the barn and the tumor has the ability to both invade and in some cases metastasize. And with the advent of new technologies, including cell-free circulating DNA, there's an increased need to understand the features that render certain tumors invasive and to really define those that are likely to be aggressive. And so with this, today I'll really speak to just several vignettes emerging from our precancer atlas of ductal carcinoma in situ, and I'll talk about how we've started to discover new molecular subgroups of disease, as well as how we've leveraged in situ proteomic profiling to define critical transitions during progression to invasive disease. I'll then go on to talk about work from our precancer atlas of familial adenopolyposis, or FAP, and I'll talk about the clonal evolution of this disease, as well as the use of single cell attack and RNA sequencing data to chart malignant transitions. And then finally, I'll wrap up by discussing some of the opportunities ahead for these massive data atlases and how we can leverage the data towards predictive models. So it goes without saying that this is really an enormous team effort. And I want to point out in advance many of the PIs that are involved in our Duke Precancer Atlas. This has been a phenomenal team to work with. They span many different disciplines, and this is really what's enabling this science going forward. So this particular atlas is led by Shelley Huang, Robert West, and Carlo Maley. And really the goals of our PCA are to understand how a ductal carcinoma, which is this non-obligate precursor, progresses to invasion. And it's worth pointing out that DCIS is a quite common diagnosis. It accounts for about 1/5 of all breast cancers, or 60,000 unique cases annually in the US alone. And of course, many of these diagnoses have been triggered by the increased use of mammographic screening, but not all DCIS will progress. And in fact, one of the key challenges has been in the management of this disease because strategies for treatment range from active surveillance to bilateral mastectomy. And obviously with that in mind, there's a critical need to improve our approach for risk stratification and to avoid the issue of either over treatment or under treatment. So the goals of our pre-cancer atlas is really to generate a multimodal atlas that applies evolutionary and ecological principles to understand the natural history and clonal evolution of DCIS, to define the molecular features associated with progression to invasive breast cancer, and ultimately to define robust strategies for risk stratification. And there's a number of many different sub-aims that fall under this category. I will speak to a few of them today, namely, the focus on understanding molecular subgroups of DCIS and their relationship to invasive breast cancer, as well as how there are changes both in the tumor compartment as well as stromal in both composition and structure during progression. So to give an overview of the pre-cancer atlas, we generated essentially 2 retrospective cohorts of patients, and each of them can have 3 possible outcomes. Either they don't recur, or they recur with a subsequent DCIS or to invasive disease. And so there's really been critical care taken to select these cases and to clinically curate them. Now, Rob, being a pathologist, has taken a meticulous efforts to demarcate tumor regions of interest and to array these on tissue microarrays. We're then able to go forward and serially section them. And an important feature of the rabbit cohort is that we perform laser capture microdissection to delineate separately tumor, so stromal regions, from DCIS. In contrast, for the TBCRC cohort, we perform macro dissection. And so these 2 cohorts have been subject to a whole array of different technologies. This includes low-pass whole genome sequencing, transcriptome sequencing. In the case of rabbit, of both the stromal and tumor compartment, they've been profiled via FISH, as well as using multiplex ion beam imaging to peer inside the tissue in situ. And one of the first questions we had with this cohort was, well, how does the copy number landscape of DCIS reflect that has been described in invasive breast cancer? And so you can see here the prevalence of copy number alterations with amplifications in red and losses in blue. This is for the primary DCIS cohort. And by and large, we recapitulate many of the known features of invasive breast cancer: 1q, amplification. We have alterations on chromosome 8q, as well as 17 and 20. And if we look at the landscape of those tumors that go on to become recurrent, we see that by and large, they're fairly similar. And so a key question that we had is, in part because we use low-pass whole genome sequencing, how does this level of genome instability compare to that's been reported in invasive disease? And so we downsampled whole genome sequencing data from the invasive breast cancer TCGA cohort, and you can see that overall the invasive cancers have a higher % genome altered, but there's relatively limited difference between DCIS that go on to recur versus those that do not. And so now with these data in hand, we set about to perform unsupervised clustering using NMF of the combined rabbit and TBCRC cohorts. What you can see below is that this delineates some 6 subgroups. And in fact, if we look in closely, we'll see that there are characteristic copy number alterations that are picked up in each of these 5 of the CNA clusters. And if we map this onto other features, we'll see that in some instances, such as cluster 1 and cluster 4, there's an enrichment for HER2, the HER2 intrinsic subgroups, and indeed overexpression of HER2. In other groups, we see more of an enrichment for luminal-like tumors and so forth. And so really with this, we're able to show that there's an ability to recover in an unsupervised fashion some of these different features. And as we'll go on to show, it turns out that some of the regions that are defined here are recurrently amplified in invasive breast cancer. And this leads us back to work from my own group some years ago in the Metabrit cohort, where we demonstrated that there's some 10 to 11 subgroups of breast cancer with defined copy number alterations. So this extends beyond the well-known HER2 group, amplified at 17q, but we see that there's other integrative subgroups that have similar high-level copy number alterations. And moreover, we've been recently able to follow up on this looking at the 20-year clinical outcomes. And what we found was really quite striking, namely that if we chart the probability of relapse across these 11 integrative subgroups, with the HER2 positive group over here on the right, and this is before the routine use of trastuzumab, we see that, of course, they have a very high risk of recurrence, but there's also 4 high risk of relapse ER positive HER2 negative groups mapping back to these copy number alterations with exceedingly high risk. In fact, their risk persists up to 2 decades after diagnosis. And so it was very intriguing to us that if we look at these copy number alterations within the DCIS cohort, what we see is that there's an enrichment indeed for some of these high risk associated copy number events, including that on 11q13. There are in fact 2 distinct groups that enrich for HER2 amplification, cluster 1 as well as cluster 4, and this is a bit of a broader amplicon. And then we see this group here with 8p12 amplification enriched in group 2, and this maps to the integrative cluster 6. So this suggests that these alterations actually occur quite early during disease progression. Now, we were also interested in how these changes map onto the intrinsic subgroups of breast cancer. And so what's shown here is the breakdown for TBCRC by controls, cases that recur to DCIS or invasive breast cancer, as well as for rabbit. And I'll point out that, of course, the TBCRC cohort was macro dissected, and this is a bit different than looking at just the tumor enriched compartment or epithelial compartment from the rabbit cohort. So there is quite a big difference in composition, and this is also attributable to differences in the cohort. But what I also want to emphasize is that it's perhaps not so simple. So if we actually ask how well we are classifying these subgroups, broken down by DCIS versus invasive disease, we'll see that mapping onto the PAM50 or intrinsic subgroup centroids, there's a far lower correlation, particularly for the basal group in DCIS and relative to invasive, but for virtually all of the subgroups. And this suggests that we're perhaps not classifying as accurately as we might have anticipated. Now, if we plot this in UMAP space, we can see that, again, there are some quite a bit of spread amongst the intrinsic subgroups. And in contrast, if we try to discover these subgroups de novo using NMF-based clustering, we find a far better stratification and separation into some 3 groups. So what do these groups look like? Well, looking at the RNA-based clusters, just showing here the heat map for the top 90 most informative genes, we see that really they're driven by the abundance of HER2 as well as ESR1 or estrogen signaling. And so, for example, we can see enrichment for HER2 in this group. In contrast, group cluster 2 is enriched for normal-like PAM50 subgroup, and then here we see the luminal-like subgroup. And if we look at the association with respect to outcome, we see that the group 2, shown here, actually has the best prognosis. This is the normal-like group, whereas the HER2 enriched group, subgroup 1, tends to have the worst prognosis. And that's consistent with some of our expectations. And these RNA-based clusters also recapitulate in the rabbit cohort. So now I want to shift gears a bit and say that in addition to investigating changes in the microenvironment via laser capture microdissection, it's also the case that we can really interrogate this quite deeply by peering into the tumor tissue in situ using MIBI-TOF. This was a technique pioneered by Mike Angelo. And for those of you that are not familiar with it, MIBI uses antibodies tagged with isotopic metal reporters read by time of flight mass spectrometry. And this allows us to simultaneously read out some 40 markers. And you can see the panel of markers that was used here for the DCIS cohort. They span both tumor features as well as myoepithelial, stromal, immune, as well as markers of immune cell regulation and functional markers. And so in work that was led by Tyler Rysam, a postdoc in Mike Angelo's lab, together with Rob West, they really set about to do a deep dive to understand how the immune microenvironment changes during progression to invasive disease. And so they took a subset of cases from the rabbit cohort. You can see that some of these progressed to develop an invasive recurrence, and others did not, and there was a total of 11 years follow-up for this cohort. And so now combining MIBI and LCM-based RNA sequencing, one can really delve into this far deeper. So here's an overview of their profiling approach. What you can see is a representative image of a DCIS tumor with a 9-marker MIBI overlay. And this shows all of the major cell lineages. You can see that this accurately matches the H&E-based image and shows really a highly multiplex view into these underlying tissue features. And from there, they're able to use single cell segmentation strategies and cellular markups to really appreciate how the cellular composition changes throughout different regions using different masking strategies. And they identified a total of 16 major subgroups shown here. They're ranked by the total abundance. And this includes both tumor cells as well as fibroblasts and myoepithelial cells and a diverse array of immune cells. So now with this spatial information preserved, you can overlay these different cellular phenotypes onto their spatial locations to start to chart tumor cell neighborhoods. And what you can see from this is that here the tumor cells in light blue are surrounded by these dark blue myoepithelial cells, but they're completely isolated from the immune cells in the stroma. And if we plot this in UMAP space, marking cells by their identity, you can see that there's this structure of isolated tumor bound by myoepithelial, and again, separated from all the stromal cells. And so this is really just a deep dive into the architecture of these precursor lesions. Now, beyond looking at the cellular features of DCIS, there's a whole array of additional biology that can be unpacked using this technology. And so in particular, MIBI allows us to look at the myoepithelial morphology and phenotype. And you can see here that in normal breast tissue, the myoepithelium is quite thick, but in DCIS, this becomes very thinned and far less cellularly dense. And you can actually quantify the density of these myoepithelial cells. Interestingly, we find that DCIS lesions that are mixed with invasive breast cancer in synchronous lesions, which may be closer to transitioning to an invasive cancer, tend to show a more similar density. And what's really intriguing is that the synchronous lesions tend to show this upregulation of phospho-S6, which may be an attempt to maintain their barrier to invasion. Now, because this myoepithelial layer is so thin and stretched out, classic image segmentations do not work here. And so they developed a unique approach in which you can apply this sort of myoepithelial masking strategy based on a pixel level classification. And this allows for phenotyping of smooth muscle actin positive myoepithelial. And then you can cluster them back into the phenotypes. And so what this reveals is that there's really high levels of vimentin. You can also see changes in cytokeratin and E-cadherin expression. Now, if we zoom into this a different way, so looking at an overlay of these different clusters, we have on the top a tumor with high E-cadherin and cytokeratin positive myoepithelium, and then on the right one that's predominated by vimentin and CD44. And we can actually start to see the MIBI image overlays here. And importantly, when we look at the top features that we're able to discriminate between women who never recur versus those who developed invasive disease after surgery for DCIS, we see that there's really this striking prominence of E-cadherin positive smooth muscle actin positive myoepithelial states. And this turns out to be amongst the most predictive. And in fact, if we quantify this in invasive cancers versus those that do not recur, we find that this is statistically significant. So in summary, what I've shown you from this quick window into the DCIS Atlas is that DCIS does not robustly map onto the intrinsic expression-based invasive breast cancer subgroups. Instead, we see that there are novel subgroups and they're predominantly driven by ER and HER2 signaling. We find that genomic copy number alterations that are associated with high risk of relapse invasive breast cancer are present in DCIS, and this is indicative of their early origins. We show using MIBI-based analysis that the tumor, myoepithelial, stroma, as well as immune compartments all change, and this is both compositional as well as phenotypic in nature along the trajectory to invasion. And importantly, a classifier that was trained based on MIBI features was able to predict DCIS that went on to progress to invasion. And so ongoing efforts within this atlas are focused on characterizing clonal evolution, bringing in whole exome sequencing, working on integrating these multimodal data types, as well as identifying additional features that are associated with recurrence. And future work will also focus on the prospective component of this atlas. So switching gears a bit, I want to spend some time thinking and talking through our efforts around the pre-cancer atlas of FAP. And here we have really focused in on how we can leverage whole genome sequencing to understand the clonal evolution of FAP. We've performed a single cell analysis charting the path towards malignant transformation. And so again, this has been a huge team effort involving many individuals at Stanford. This is led by Mike Snyder as well as Jim Ford. And there are as a strong computational component, as well as our surgical and pathology colleagues. So FAP has been a tremendous model of colorectal cancer initiation. And that's in part because while it is an autosomal dominant condition caused by mutation in the APC gene, and although it accounts for only 1% of all CRCs, we know that APC mutations are the initiating event in over 80% of sporadic colon cancers. And FAP patients invariably develop hundreds of polyps and they undergo prophylactic colectomy, making it really an ideal model to capture different polyps at different stages of progression within a defined genetic background, within the same individual even. And it's also known that FAP phenotypes can vary quite extensively, and that they're only partially attributable to differences in the specific germline APC mutation. And this suggests that there are modifying genetic events, but also potentially environmental factors. And this, of course, has implications for intervention. So what were the goals of this atlas? Well, we really sought, again, to deploy evolutionary principles and data analytics to understand the natural history and clonal evolution of FAP. We wanted to delineate features that are associated with progression to invasive disease, and ultimately to define these modifiers of risks and strategies that might enable us to therapeutically intervene. And again, this has a number of sub-goals. This includes charting the evolutionary dynamics of tumor initiation progression, which I'll speak to, as well as identifying molecular changes during the transition to invasion. And ultimately, we're also very interested in understanding how these changes in FAP differ to those observed in sporadic colorectal cancers, for which there has been far more extensive efforts to profile the somatic landscape. So within this atlas effort, we've really undertaken a pretty comprehensive approach. We are interested in characterizing mucosa or normal regions of the colon, those that are benign, polyps that are lacking dysplasia, those with dysplasia, as well as when they arise and are not captured beforehand, the paired carcinomas. And of course, we're hoping to avoid this with the prophylactic colectomies. So this is just showing a schema of how we go about sampling, and it gives you some idea that we can actually record spatial information along the entire colon. And so really have this unique opportunity to understand in space as well as time how these polyps develop, why some of them progress and others do not. And we've brought to bear a very large compendium of different omics. This includes whole genome sequencing, which I'll speak to today, methylation profiling via whole genome bisulfite sequencing, RNA-seq, single cell RNA and ATAC-seq, I'll also touch on this, proteomics, metabolomics, and lipidomics. So really bringing the entire armamentum of omes to bear to understand this disease. So, starting first with the whole genome sequencing data, and this is work led by Aaron Horning, a joint postdoc in Mike Snyder in my lab, we took a deep dive into the landscape of somatic alterations that arise on this backbone of germline APC events. So what is shown here is across 6 patients with FAP. You can see the prevalence of different alterations that accrue. Clearly, there's common alterations in the Wnt signaling pathway, as well as RAS, TP53, and Notch. And these are largely drivers taken from the Cancer Genome Atlas and that are known to be somatically altered in colorectal cancers. You can see that there's also a whole array of somatic copy number alterations that occur. And in fact, because we've ordered samples here where increasing size of the lesion is indicated, we see that there's an increase in somatic copy number alterations with increasing size of the polyp. And the patterns of somatic alteration do not necessarily track specifically with location in the colon. Now, almost all of the samples that we've seen have both APC and KRAS mutations. This occurs in even some of the smaller samples. And importantly, we can note that APC and KRAS are individually associated with stage. So this is a second hit, a somatic alteration in APC. We see the same for KRAS. And in fact, this is also true for combinations of these common colorectal cancer driver genes. Now, we've been annotating these colons quite exquisitely. It takes a real team effort to go in and actually do this, just given the sheer size. You can see some of these are 10 centimeters long. And what we are able to do then is to digitally convert the spatial information into a polyp density map, if you will, and to correlate that with molecular profiles. And this is really important because understanding how the space plays a role during tumor development and the location within the polyp, whether it's ascending, transverse, or descending, is quite important. In fact, we know that there's different biology associated with cancers that arise in different regions of the colon. So one thing that's quite apparent when we do this study, really looking for the first time at exquisite resolutions, exquisite spatial resolution, is that if we chart and the patterns of somatic alteration, this is looking at private mutations and those that are shared, that are predicted to be pathogenic with a CAD score greater than 20. And we look at these, how they map across the location of the colon. What's clear is that there are mutations that are shared, identical mutation found in very disparate regions of the colon. And this is really quite intriguing because it begs the question, how do these mutations spread in that manner? We can look at this a little bit more deeply, in this case now, reconstructing the sample phylogeny for the same patient, A001. And what you can see from this plot is that, first of all, there's extensive subclonal sharing. We see that this occurs across different regions of the colon, indicated here on the branches with different colors. It occurs also across different stages of disease, as indicated here by the terminal node, whether it's mucosa, benign, dysplastic, or the adenocarcinoma. And also there's some very long branches indicating the acquisition of specific private events that are accruing within a particular lineage. And so this begs the question, how does this subclonal sharing occur? When does it occur? And if we, just to convince us that in fact, mutations are not tracking with space within the colon, we can plot the Jacquard similarity index for patient A001 and A002. This is a measure of subclonal sharing, and we see that it really doesn't correlate with Euclidean distance whatsoever. We could also look at this a different way and look at the interactions that are observed, again, plotting Jacquard similarity according to increasing stage for interactions between mucosal regions, mucosal and benign, mucosal and dysplastic, and so forth. And what we tend to see across many of the patients in this cohort is that there's a sort of, with increasing stage, a decrease in the extent of sharing that's observed. And really what this leads us to is a model in which we envision that early during tumor development, it's very easy for these polyps to spread, for mutations to spread. And this is perhaps also influenced by field cancerization. And so we have this subclonal intermixing within the small fetal colon in these patients who harbor an APC mutation at birth. And these patterns of spread tend to expand as the gut increases in size and as the individual develops. And so what's really intriguing is that we can also simulate this process, drawing on some of the evolutionary tools we've developed for colorectal cancer in the past. And this again indicates that it's very likely that most of these polyps are polyclonal in origin, and this is consistent again with this early spreading event. So shifting gears a little bit more and coming back to single cell analyses that we've been able to pursue on this cohort in work that was led by Winston Becker and Stephanie Nevins to graduate students together with Mike Snyder and Will Greenleaf, we've been able to go in and survey really at unprecedented resolution regions of normal, unaffected mucosa, adenomas, and carcinomas. This bar plot shows how the specimen was obtained, whether from colectomy, autopsy, or otherwise, and whether the patient saw any preoperative radiation therapy. So for a total of 48 polyps, 27 normal tissues, and 6 cancers, we were able to collect over 200,000 single cell transcriptomes and 450,000 single cell chromatin accessibility profiles. And what we see when we do this is essentially that we can generate a UMAP representation for these different stages of disease, looking first at the RNA-seq and second at the attack-seq. And we cluster cells based on where they were isolated from. And what's clear is that these stromal and immune cells from all cell types tend to cluster together, while the epithelial cell clustering is driven by the state of disease, with cancer down here and adenomas over here. And this is true for both of these molecular profiling technologies. And importantly, we can now leverage the fact that these representations can be mapped onto cell annotations of immune cells from all different samples. And so when we annotate both the single cell RNA and attack-seq data, we can then go in and quantify gene activity scores, as well as the expression of marker genes. And so what we see here is that there's a whole host of markers of immune expression that vary really quite dramatically across these different populations. These include markers such as FOXP3 and GATA2. Now, what was intriguing to us is that as we sort of zoomed in and looked at, particularly at the carcinomas, we noticed that there was a cluster of T cells that was uniquely present, we believe, in the colorectal cancers. And in fact, this population was supported by multiple lines of evidence. So if we look again using gene activity scores based on the attack-seq, we can see that there's markers of T cell activity, and that the accessibility of motifs of transcription factors such as CTLA4 and TOX, that they were labeled as being exhausted in these attack-seq data sets and really mapped back to this population. And moreover, when we turned back to another single cell RNA-seq data set from basal cell carcinoma, where we had similarly defined exhausted T cell populations, we again see this overlap and enrichment. And so really we believe that this exhausted T cell population is unique to the colorectal cancers. Now, again, using this ability to score gene activity from the attack-seq data, we went in and annotated the various populations of stromal cells that are present. And I just want to point out that this includes markers such as Wnt5b, Wnt2b, and arspondin3. And we can now map these across really the entire data set. And intriguingly, what's apparent from this is that there's really unique populations, again, in this case of cancer-associated fibroblasts, that are exclusively present in the carcinomas. Now, there is this sort of pre-CAF-like population that is coming up also in the polyp population. And so taking it a step further with the attack-seq data and mapping this back onto RNA, we're able to start to identify regulatory elements that are correlated with gene expression changes. And so what's shown here are genomic tracks for accessibility around the Wnt2 and around RUNX1. And what you can see is that there's the greatest accessibility around Wnt2, which is a CAF marker that activates Wnt. And this is really present in the cancer-associated fibroblasts as well as the pre-CAF population. And when we look at the RUNX1 locus, what we start to see is that there are transcription factors around the TSSs, again, in the cancer-associated fibroblasts and the pre-CAFs. So this enables us to now start to systematically define transcription factors that may be driving gene expression in these stromal cell populations. And so we can look using Chrome bar and plot the deviation of the Z-score. So this essentially depicts the transcription factor motif activity. And what's clear from this, sort of moving along, looking at RNA on the top and accessibility on the bottom, is that there are particular motifs for both RUNX1 and RUNX2, and they tend to have actually similar patterns. But we believe that it's RUNX1 that is actually expressed within the CAFs and is likely responsible for the increased accessibility of RUNX1 motifs within CAFs. So taking all of this data and really combining it, we're able to also project the polyp data onto a normal epithelial manifold. And when we do this, what we start to observe is that there's an enrichment for stem cells, specifically within the polyps, and this continues to increase to invasion. And so we can actually see how this looks first here for the stem cell markers, but we also find quite strikingly that it's not only the stem cells that seem to increase in terms of the percent stem cell fraction that's present along this continuum to malignancy, but we also see that there are many other cell types, including enterocytes, as well as exhausted T cells, that tend to show this same trajectory along the continuum to malignancy. And so we can also leverage these data to now come back and ask the question, well, if we take the RNA-seq data and ask about differential expression, what's up and what's down, again, along the continuum to malignancy, we can see that there's significant numbers of genes that are altered in each sample relative to all unaffected samples. And this heat map here illustrates the genes that were differentially expressed and where samples are ordered again along the continuum to malignancy. And so these are log2 fold changes. And what's intriguing is that we see that for the GPX2 gene, this seems to be really enriched in stem cell-like markers. And we see that this increases when we compare relative to unaffected normal. And importantly, GPX2 is thought to control oxidative stress within the colorectal cancer microenvironment. But it's not the only marker that changes. In fact, we could find related patterns for many other stem cell marker genes, such as LREG1, RPL13. And so again, this seems to suggest that we're observing the selection of genes in stem-like cells, and that this is changing along the continuum to malignancy. So we can do the same kind of analysis, but from a regulatory perspective, now leveraging the ATAC-seq data. And so again, looking at the number of significant differential peaks for each sample relative to all unaffected samples, again, ordered along this continuum. What's shown here is a heat map of all peaks that were significantly differentially expressed. And again, the samples are ordered along the continuum here. And we can now perform a hypergeometric test to identify transcription factor motifs that are enriched here, and we can cluster them using k-means. And what stands out from this quite immediately is that there are changes in the expression of AS-LC2 as well as HNF and HNF4A, and this tends to again be enriched in the stem-like population relative to unaffected samples. And so it seems to follow along this trajectory. And so we can see these patterns mirroring what we had demonstrated before. And so what I've shown you here is that really leveraging whole genome sequencing across multiple polyps from the same donor, we find evidence for extensive subclonal mutational sharing. This occurs independent of physical distance. Independent mutations are also common and sometimes give rise to these long branching trees within a particular location. And through simulation studies, we're able to demonstrate that really the patterns of subclonal sharing that we observe tend to be consistent with the model in which the polyps are of polyclonal origin. And this suggests intriguing patterns of how mutations are shared during the expansion of and progression to invasion. Now, the polyp and colorectal cancer cells both exhibited a stem cell-like phenotype, and we observe a continuum of both epigenetic and transcriptional changes during malignant progression. And I didn't have time to talk about this, but this is also mirrored at the DNA methylation level. And we find that advanced polyps tend to show these increasing numbers of stem-like cells as well as regulatory T cells. In contrast, CRCs exhibit evidence for T cell exhaustion and RUNX1 regulation of CAFs, as well as increasing accessibility associated with HNF4A motifs. And so ongoing efforts within this pre-cancer atlas have been focused on integrating across the many other OMEs that I didn't have time to share with you today. And we're also very interested in building out our evolutionary analyses further and surveying additional prospectively collected cases. So I just want to spend a few minutes discussing some of the opportunities that I see ahead. And want to point out that there's just a treasure trove of data that is emerging from these atlases. And as Sean indicated, the first data release will be coming soon. This includes not only the atlases that I've described that are charting progression from pre-malignancy to malignancy, but also comparisons of primary tumors and metastases, including some that have been sampled pre and post-treatment. And the measurements, as I illustrated, differ to some extent across the different atlases, both as a function of what's practical, but also the specific questions that are of highest priority. And really, I've described a little bit of our work to begin to delineate cell types, to understand gene regulatory programs, as well as cell-cell interactions and their cellular structure. However, I think the greatest task really lies ahead of us. And that's now that we're having these well-curated atlases that are searchable and findable and computable, how do we take this to the next level? How do we actually leverage this to predict response or to risk stratify? And I think that there's going to be a huge opportunity for really bringing to bear multi-scale models that bridge the molecular level all the way up to the cellular level. There's obviously great opportunity for thinking about different ways to model regulatory programs and to leverage the longitudinal data that is inherent to these. And I think that is really fills a missing void in the field, because most of our data thus far has been a single snapshot in time. And perhaps I'm biased, but I also believe that there's a real opportunity to now go back to some of these models of tumor evolution that we and others have developed and start to embed with more rigor the microenvironmental and spatial information now that we have a window into tissue architecture. And so we're very excited about this. And I would also just say that I think that we really need to be thinking about these atlases through the lens of evolution. And work from Carlo Maili and many others had issued a consensus statement on this a few years ago, advocating that we could leverage atlas data such as this to define both an evolutionary index of how cells within a tumor are evolving, but also to account for their ecology. And this is critical as we bring to bear new spatially resolved approaches. It's essential that we study the tumor cells within their native microenvironment and cellular context. And we also have outlined some perspectives on this in work that came out of an NCI workshop on tumor evolution, and really highlighted some of the parameters that we think might be measurable using these technologies, both evolutionary and ecological. And so I think a frontier ahead will be to really start to measure these both within a tissue type, across progression, but also across different tumor types to learn common principles, because inherently this is an evolutionary process. And so I want to close by once again thanking my many collaborators and the people who have made this happen within the DCIS Atlas team, members from Duke, Stanford, ASU, WashU, Dana-Farber, and the ICR. And it's been a tremendous team effort to really bring this to fruition. And we're excited about the work ahead as well. And I want to also thank the members of the FAP Atlas, including those from the Snyder Lab, Ford Lab, Greenleaf, and Kanjaji Lab. And of course, all of the NCI program staff who have supported these efforts along the way. And I'd be very happy to take any questions.
Great. Thanks to Christina for such a powerful and clear presentation. We want to thank you. And we have received 10 questions through the Q&A box. And there are a few of them about MIBI. So let's start with MIBI. And so one question from Sophie is that what's the difference between MIBI and Hyperion?
Yeah, so I should say that I guess I should have outlined that there's actually many different spatial imaging modalities, and I think many of these are being utilized by different atlas efforts. What is unique to MIBI, and this was the technology that I outlined, developed by Mike Angelo's lab and Gary Nolan's lab at Stanford, is the use of these heavy metal isotope tags to then coupled with the time of flight mass spectrometry to image directly within the tissue. And so there's sort of many flavors of this that differ subtly depending on resolution and the exact approach to how these digital signals are read out and quantified.
Mm-hmm. Great, thank you. The second question related to MIBI is from Khalid. Can MIBI be applied to analyze mouse tumor?
Mouse tumors. That's a great question. So I think provided that you have the appropriate antibodies, yes. You know, a key component for any of these assays is that you have to really do a large degree of antibody validation and confirmation, first making sure that, you know, it works, that it's specific, but also that it works well in your tissue type. And that's obviously important for FFPE tissue, which maybe really works seamlessly with, but provided you have appropriate antibodies, this should be feasible. And just to say that I think, you know, antibody validation across the board has been an important component of efforts going on within the HTAN effort. So wanting to be able to share the use of similar reagents so that we can actually assimilate these readouts is going to be really important.
Mm-hmm. Great, thanks. Christina, you showed beautifully the myoepithelial layer using MIBI. So the question is there a relationship between recurrence and the thickness of the myoepithelial layer?
Right. So actually, that's a wonderful question, and I didn't have time to get into that in too much detail. I would point out that there is a preprint posted on this work from the Angelo and West labs, and in fact, they were able to identify many different features. I believe they looked at up to 1,000 features in total to ask whether spatial contributions, structural contributions, and other molecular readouts contributed to the prediction. And I believe that the thickness was one of those features, but this was a multimodal model, and I think that one of the top features that was illuminated there was the sort of myoepithelium E-coherent frequency. But there's also other features that include structural components such as variation in collagen fiber alignment and the percentage of the duct that's actually covered by the myoepithelium, and many others. And so I think, you know, in total, what's really exciting about this is that these models that can be trained on MIBI features are not specific to one sort of readout. So you could have readouts of cell assignment, of phenotypic or functional subsetting. You have these structural features, which includes what you just asked about, but there's also the spatial relationships and the cell-to-cell neighborhoods. And it appears that amongst, you know, besides myoepithelium being amongst the top, that there's really a predominance of spatially resolved features that are driving this. And so I guess, you know, an interesting question going forward that I think many of us might have, and I don't have the answer to, is, you know, how many of these features do you actually need to reliably classify? And I think there'll be a lot of work, you know, investing in this in the future. And of course, it'll be important to assemble additional validation cohorts.
Great. The next question is from Hala. And how do you distinguish tumor stroma from the normal tissue invasion, such as fibrous tissue?
Fibrous tissue, right? So So perhaps you might recall that one of the opportunities that we have here using all these different modalities is to really use different cell markers to define different cell lineages and categories. So with reference to the MIBI data, I'm not actually sure which data set is being referred to here, but from the MIBI data, a total of 16 different major cell types were identified. And these are using features. So in the case of spatial imaging, you can obviously go in and you have the histology and spatial representation of the tissue that you're looking at. And that provides some sort of orthogonal check on where these features are derived from. And you might have recalled some of the images that were shown that really very clearly delineate the sort of region of DCIS with the stroma surrounding this. And so I think this is a benefit of having the spatially resolved approaches, is that we can actually see where within the tissue those different features are coming from. Now, with the I'm not entirely sure which data set you're talking about, but I would say that, we sort of take a different set of approaches when we turn to having single cell dissociative data. So whether this is ATAC or RNA-seq. And in this case, we were able to project the data onto manifolds from both unaffected colon as well as the adenoma and carcinoma populations. And here, of course, we have, whole transcriptome, whole readouts, and so are able to use combinations of these different markers to annotate and define cell populations. And there's a whole process and actually efforts within the HTAN that go into how these annotations are done and can be transferred across different populations. And I think that's a really important area. So it of course depends a little bit on your on the resolution of your data.
There are still questions coming in, but due to the interest of time, this will be the last question, Christina, for you to answer. This question is from Scott. Are you collecting sequencing data such as VDJ as part of the exercise, especially through single-cell RNA-seq?
That's a great question. We do have plans to do that. We have not done that so far. So that's something certainly that's been discussed for different projects. I would say that so far in our experience, this tends to work better with fresh material than with formalin fixed material. So there's a consideration there. I think depending again on the source of tissue for these different atlases, in some cases we're dealing with very limited amounts of material. So we often need to prioritize the assays that are selected. In contrast, if we are, working in the setting of colon where we have an entire colectomy sample, this is usually not a limiting consideration. So we hope to have some of those data in the future. And I would say that I would anticipate that some of the other atlases are doing this as well, particularly those that are studying, for example, response to immunotherapies.
Thank you so much. Thank you, Christina, for answering those wonderful questions, and thanks to people who asked insightful questions, and remaining questions will be answered later through online. And now I want to turn this to Xiang.
Thank you, Lee. So I just want to reiterate the thanks to Christina and to Lee and to all the question askers for a great presentation and a great discussion. And then I want to finish up by just announcing next month's Cancer Moonshot Seminar Series, which will be given by Carl June from the University of Pennsylvania, and it'll be updates in CAR T cell therapies. So please, everyone, thanks for attending, and hope to see you again next month.